Hello everyone, myself Navya and my teammate Manovnya are here to tell you about raw materials and fermentation media. What is fermentation? It is defined as a process in which chemical changes occur in organic substrate through action of enzymes produced by microorganisms. Raw materials required for fermentation are carbon source, nitrogen source, fat and hydrocarbons, ammonia, nitrate, nitrogen, and phosphate source. Carbon source. It is required for all biosynthesis leading to reproduction, product formation, and cell maintenance. Example molasses, malted barley. Nitrogen source include amino acids, proteins, and urea, corn steep liquor, yeast extract, peptones, and soya bean meal. Microbes require mineral elements for growth and metabolism. In many media, magnesium, sulfur, calcium, chlorine are essential components and must be added. Chelators. Many media can't be prepared without precipitation during autoclaving. So chelating agents are added to form complex with metal ions. Example EDTA citric acid. Vitamins and growth factors. Bacteria can synthesize all vitamins from basic elements, and for some supplements must be added to fermentation media for good growth. Water. It is not only major component of all media, but it is important for ancillary services like heating, cooling, rinsing, cleaning, etc. Oxygen. Depending upon amount of O2 required by organism, it may be supplied in form of air and occasionally as pure oxygen when requirements are higher. Antifoams. Foaming is largely due to media proteins that become attached to air broth interface where they denature to form stable foam. Antifoams reduce foam formation during fermentation.
Coming to the fermentation media is very important for high yield of production, and it provides nutrients and energy for growth of microorganisms. It also provides substrate for product synthesis in a fermenter. Fermentation media consists of major components such as carbon source, nitrogen source, and minor components such as inorganic salts, growth factors, vitamins, buffers, antifoaming agents, etc. Coming to types of fermentation media, synthetic media. In this media, concentration of nutrients can be controlled and the yield of the product is analyzed. It can control both growth and yield of production. With the help of radioisotope labeling technique, we can determine the main ingredients that get used up to create final desired product. Crude media. It is generally used on an industrial scale for fermentation process. It gives high yield of product and contain undefined source of ingredients. Those ingredients in crude media are inorganic nutrients, which contain inorganic salts along with carbon source, and carbon source simple to complex carbohydrates can be added to the media. Nitrogen source, salts of urea, ammonia, and nitrate can be used as nitrogen source. When fermentation organisms are proteolytic in nature, animals and plant raw materials is used. Growth factors. Crude media constituents provide enough amount of growth factors, so no extra addition of growth factors is needed. Example yeast extract. Precursors. They are generally present in a media as crude constituents, and precursors are added as they get incorporated into the molecules of product without bringing any kind of change. Buffers. Buffers are used to control drastic changes in pH. Thank you.